Thank you very much for inviting me. I'm quite excited to present among this audience now, because as you said, I actually work in a little bit of a different field in bottom-up synthetic biology. And with my own group, I study more how the immune system works and how we can tune it. But I actually thought when getting the invitation to present here that we have been doing some work some years ago and now keep on doing this as kind of a side project that might be very relevant for this project. That is work that we published already in 2020 in Small, and it concerns the bottom-up assembly of functional intracellular synthetic organelles. So maybe I start off by telling you a little bit more about what bottom-up synthetic biology actually does. This is a field of research where we try to understand life on Earth, how it works, how it emerged, and how we can actually tune it in a therapeutic way by reducing the complexity of living systems. So, for instance, we would be looking at a living cell as sketched here, not as a super complex but tiny system filled with many, many different molecules, proteins, sugars, DNAs, and so on. But we would see it as a module-based system that we can reduce in complexity, for instance, by starting to take off bits and pieces in there. That is a conventional top-down approach to synthetic biology. In bottom-up synthetic biology, we do the other way around. We start with the individual molecules or the building parts for these systems, and we try to assemble them in a way to create a living system or something lifelike with a very reduced but defined complexity. What we use for this in many, many cases is actually a microfluidic-based assembly pipeline. So we use droplet-based microfluidics to assemble tiny compartments that act as synthetic or artificial cell components. They're in the size of a cell, and we can, with the help of microfluidics, actually engineer pretty precisely what we do with these tiny compartments. We have all kinds of different modules. We can fuse individual compartments with each other. We can mix the compartments inside such a, the molecules inside such a compartment in a very controlled manner. We can sort them. We can inject new molecules into them, and we can split them to induce something like an artificial cell division. So all this has been designed in order to have a very controlled handle on the systems that we produce. And what is the typical system that we produce at the end? Well, this is a lipid vesicle. That is essentially a lipid membrane encoded compartment that serves as a proxy for an artificial cell. So talking about artificial cells, yeah, that is interesting, but it's not getting much closer to the actual topic of this presentation, which is synthetic organelles. However, when we were using these type of synthetic cells and interfacing with natural living cells, human cells, we started to note a phenomenon that we decided could be very interesting actually for the creation of synthetic organelles. And this is that in some instances, we could put a synthetic cell here, this small yellowish compartment that you see, in close proximity with cells, and there would be merely an attachment, no other real interactions. But in some cases, we noticed by engineering the physiochemical properties of these compartments an increased endocytotic activity. And having a membrane-based compartment, such a small vesicle, endocytosed into a living cell is something that's very reminiscent of a process that happened, or presumably happened, many, many years ago on Earth, which is the creation of eukaryotic cells from prokaryotic cells by engulfment of vesicle-based or membrane-enclosed compartments such as bacteria. So the whole concept of the approach I would like to present to you is essentially mimicking this exact process. It's taking natural cells, like you see here on the right, that have compartments, a nucleus, for instance, other smaller compartments and organelles such as mitochondria, and installing GUV-based synthetic organelles inside. These organelles could be defined with the help of these microfluidic systems to perform any kind of functions that we install in them, to have any kind of a structure that we want to have. But the important thing is that they're actually introduced into the cells by their own. It's a self-regulatory process. And because we include molecules into the inner compartments of these synthetic organelles that allows them to be not degraded within the cells, this is actually a system that is stable for many days, up to several weeks. So it's a stable incorporation of an artificial membrane enclosed compartment into a living cell system. And I want to present you 3 approaches of what we could do in this case. First one is a very simple one. It's merely an organelle, I would argue. It's rather a lipid compartment that contains a catalyzed enzyme, essentially an enzyme able to decompose hydrogen peroxide into water and oxygen. These compartments can be produced, obviously, and we can incorporate them into living cells. And then we can study how much reactive oxygen species are actually produced within the cells with a fluorescent marker called cell ROX, which is sensitive to oxidation and glows under oxidation. This means when we stress such a cell layer, this is a microscopy image here, with menandrionine, which is a producer of hydrogen peroxide, we can actually see that the amount of fluorescence from the cell ROX green probe actually is increased, and we quantify this, we can see, well, eventually an increase. However, if we introduce these catalyzed containing peroxisome mimicking synthetic organelles, we can reduce the amount of reactive oxygen species introduced into the cells, again, almost back to control levels. So this is a simple proof of concept, I would say, of how an enzyme can work within the synthetic organelles in the cells and sustain their physiology. A little bit more of a complex system and also a more responsive system is based on mitochondria, where we try to mimic the calcium storage capabilities of mitochondria. Mitochondria are mostly known for their role in metabolism and energy provision within the cells, but they're actually also, together with the ER, the sites where most of the calcium ions are stored within the cell. They're released under physiological conditions. Sorry, this video should come first. So if we look over time on a cell layer, we measure the amount of calcium that is within the cytoplasm, we can actually see that spontaneously some cells start to fire kind of a calcium action potential within the cell layer. And this is an important way of how cells communicate. And we thought about mimicking these type of communication and intracellular calcium release by incorporating into our synthetic organelles calcium ions that are caged with a light-sensitive EGTA version. So effectively, that under light irradiation, this calcium would be released and passively diffused through the membrane into the cytosol of the cell, and by this trigger subsequent signaling in other cells within the cell layer. So this is how it looks. That's the concept. If we now look at such a cell that has our synthetic organelles inside, these are here marked in blue, and we play over time the video for this quantification in the lower back and in the lower part of this image, you can see that in the times where we actually fire UV light onto the cells, we get spontaneous release of the calcium from within the organelles into the cytosol. And this, in turn, increases the calcium signal in a cell that's just close by this actually triggered cell in here. So it's a very primitive form of interrogating calcium homeostasis in a synthetic organelle-based format. Well, both of these examples that I showed you now are things that our cells, human cells, animal cells, and many other cells in the tree of life actually have, but they're not new to the cell. They don't provide any additional functionality that one would like to introduce, for instance, when talking about space biology. However, with the same concept, we can now transplant effectively the concepts from other organelles that other types of living organisms, such as magnetic sensitive bacteria, came up into human cells, and by this provide them with new functionalities, new sensors, or a new equipment to cope with other types of stresses. So in this case, and to realize that concept, we produce synthetic magnetosomes. So essentially mimicking these structures that you would find in bacteria that allows them to sense magnetic fields and orientate themselves according to the Earth magnetic field. Again, very same concept as I introduced for the other ones. We produce synthetic organelles that in this case have magnetic or magnetosensitive nanoparticles in them. Once they would be placed close to a magnetic field, they would actually align themselves, exert a force from within the cells, and then induce a magnetotactic movement from these essentially non-magnetosensitive cells to the magnet. How does it look like? Well, this is our compartment with the magnetic particles inside, you can see that we can deviate them with a magnetic field from outside. So we have a direct responsive system in there. The synthetic organelles are closely linked with the actin cytoskeleton, as you see on the lower left. Yeah, so the cells actually, once they're introduced into their inner cytoplasmic states, they tightly connect their actin cytoskeleton with these synthetic organelles, which allows them to, and here's a magnification of this whole thing, actually sense the directionality of a magnetic field that is close by. So all the magnetosomes, here you can see 4 individual ones, would align within the cell according to the space. We can also see that on the ultrastructural level, that they actually start to push against the inner membrane of the synthetic organelle. And if we look on cells that are plated in a standard regular well plate, this is a whole scan of them, either with synthetic magnetosomes or without, in a magnetic field, you can actually see that all the cells, or most of the cells, would start to migrate to the right side of this well plate where the magnet has been placed. This is a quantification of this. And essentially what it shows is that we are able to install a magnetotactic sense in the form of an organelle into living animal and human cells. So to conclude what I just showed you, I hope that I was able to convince you that organelle mimicry is actually a way to go when we try to understand the role and the function of organelles a little bit better, when we want to also introduce additional or new functionalities into living cells. For this, or following this concept, we can enhance the therapeutic usage of some cells, but we can also install completely new functionalities that might be relevant for things like space travel, where increased reactive oxygen or oxidative stress would be placed onto this living systems, or we can introduce completely new types of functionality, such as the magnetotactic sense that I showed you. We have been working now on photosynthetic systems, so essentially introducing a light harvesting system in the form of a synthetic organelle into human cells. And all these toolbox of different types of synthetic organelles that could be introduced into living cells, I think could significantly expand the functionality of our own cells when we prepare for extraterrestrial life. With this, I would like to thank all the people from my group, people that were involved, obviously the co-authors of this paper. I would like to thank you for joining the session, and I'm happy to answer questions. Thank you very much.